Hello, hope you're all doing well. We are going to be talking about Parkinson's disease and whether it begins in the gut. My name is J-Neuro. Alright, so as we all know, Parkinson's disease is a progressive neurodegenerative disease that affects about 1 to 2 people per thousand. And it seems that the prevalence of this disease is increasing, as it seems to affect about 1% of those who are 60 and over, usually in the form of idiopathic PD. So, a brief historical background on this disease. It was first discovered by a man named James Parkinson, and he essentially named it shaking palsy when he discovered it. And he saw that some of his patients who came complaining about this before they showcased this hallmark motor symptoms were complaining about constipation. So they were the first to he was kind of the first to state that there was some link between GI issues and the eventual development of this disease. But as we know, this is not really covered when you think about Parkinson's disease, because usually the main idea is that there's this degeneration within the brain, and because of that degeneration, the motor symptoms come, and that's what we need to focus on. We need to focus on how we can ameliorate these motor symptoms and making sure the brain is all well and good. But that just it essentially just removes from the fact that there's more to it than just one system, essentially, which brings me to my next point here, that states that it's possible that some environmental toxins may cause the increase of a certain protein called Alpha-Synuclein, and this causes PD by essentially hijacking some areas within the gut that are called vagus, a nerve specifically called vagus nerve. So through vagal afferents, which are the nerves that are traveling between certain target organs, such as like the gut, and travel all the way to the brain. So they hijack these afferents, and then through that sequence would essentially just invade the brain and then cause the pathogenesis that we see in PD. But what is this vagus nerve? So vagus nerve is cranial nerve number 10. It regulates internal organ functions such as breathing, respiration, digestion, heart rate. It also helps with reflex actions, just coughing, sneezing, swallowing as well. It seems to be very prominent when it comes to just these all autonomic functions. And these vagal afferents, as I was talking to earlier, would be these nerves that are just basically connecting to areas within these target organs, and then the afferents would be just traveling to the brain. And the main highway, or the main organ by which this vagus nerve talks to the brain is called the dorsomotor nucleus, or the vagus nerve, which is situated around the brainstem. So it would be around this area right here, around the base, or the base of the head, essentially. Yeah. So what is this brain-gut connection when it comes to PD? So as I stated earlier, James Parkinson postulated that there is some GI disturbances that he noticed for within the patients that came in before eventually showcasing the hallmark of Parkinson's disease. So obviously, when we think about Parkinson's disease, most the idea is that there's some disturbance that occurs within the brain, and that disturbance is in the form of substantia nigra neuron loss, that's causing dopaminergic neuron loss, that's causing the symptoms of bradykinesia, rigidity, and tremor that are very, very common in patients who have Parkinson's disease. But this is just one story of how this disease starts. When it comes to idiopathic PD, which is a type of Parkinson's disease that the cause of which is unknown, the idea it doesn't really rest, because we see the same thing where there's a lot of degeneration of the neurons, but the idea of where this happens is just unknown. So it's not right to just leave it to that specific area of the brain. Another thing here is that PD patients only often when they experience the motor symptoms early on, they seem to also complain about certain GI issues that they're experiencing, and this could be due to a accumulation of a protein called Alpha-Synuclein, which is not just found within the brain. It's found within the nervous system all throughout the body. Not all throughout the body, but more specifically all throughout the brain and as well as the gut. So this could be through the infiltration of the enteric nervous system, which is another nervous system that is kind of more associated with the autonomic nervous system, and it's more associated with peristalsis, or rather moving food from the mouth all the way to the anus. So peristaltic movement is kind of just making sure that when you swallow your food, it's an autonomic movement that makes sure that food gets to your stomach and then to your intestine, so it can be absorbed, and then to your anus. That's what the enteric nervous system helps with. So it's postulated that these alpha-synuclein aggregates could begin within this area, and through vagal afferents go all the way to the brain, essentially. There was this other study that actually looked at children for about 9 years, and these researchers they biopsied the guts of these children who complained of bacterial and viral infections. Bacterial infections could be such things as E. coli or a salmonella infection, and they found that if there was a repeated acute, they hypothesized that if there's a repeated acute infection, there was an increase. This was associated with an increase of alpha-synuclein aggregation. And furthermore, when they biopsied, when they removed when they got samples of this children's gut lumen, essentially, or what was essentially causing the infection, they found that there was an increase of alpha-synuclein within this sample. And when they introduced immune cells, they noticed that the immune cells were very much more attracted to that. So when you think about this sequence happening within the gut, you can understand that there's a lot of inflammation that occurs when alpha-synuclein aggregation occurs. It would essentially cause signals of immune system infiltration. Immune system infiltration will cause even more inflammation, and this inflammation can be more disastrous, causing more symptoms that are more associated with gastrointestinal issues. But what's more important here is that these researchers noticed that there was an increase in alpha-synuclein when these children gained these infections. So here it's just essentially saying that there is a connection here between this important protein and Parkinson's disease when it comes to associating it with certain gut infections. But rather, before we talked about the alpha-synuclein, we have to understand whether there's some sort of link between the dysbiosis that occurs and whether this dysbiosis is something that also leads to PD. So another study that I also found that looked at certain population of patients who had PD, and then kind of extracted the type of bacteria by collecting poop samples. Found that they had a decrease in this beneficial bacteria called Clostridium coccydus, Leptin, and Bacteroides fragilis, which are commensal bacteria that would essentially protect the gut by promoting its thickening, thus preventing leaky gut as well as such disorders as IBS, which is another symptom that was reported with people who have Parkinson's disease. They tend to report having an increase in just gastrointestinal issues. Inflammatory bowel syndrome was one of them, very common. But from these studies, they found that there wasn't really a specific bacterium that could have been linked, or a lack thereof of a bacteria that could have been linked to causing PD. But what they concluded was just that people who have Parkinson's disease tend to have a decrease in diversity of the microbiota, and also a decrease in bacteria that produce butyrate. Butyrate is a very important metabolite that bacteria produce. And what it usually does, it's a short-chain fatty acid, and what it does is just it promotes the thickening of the gut so as to prevent toxins from entering circulation. So and also maintaining the diversity as well. But from this, it's very easy to conclude that it's if you don't have good diversity, your gut can be really. If you don't have good diversity, that always obviously is linked with a disruption in gut, in having a good in having good gut health, right? So it has been associated with if you have good diverse culture, you're more likely to have you're more likely to be healthy. So it's hard to conclude that not having a good bacterium is what links to having Parkinson's disease, because generally not having a good bacterial culture is not good for your health in general, because you can gain a lot of disturbances in your, you can gain a lot of disturbances within your, because of this, because having a disturbed gut microbiome can cause such a decrease in this disorder. So it's important that that point is kind of that point is justified, or that point is emphasized more. It's hard to link one bacterium to the prognosis of Parkinson's disease. Nonetheless, another researcher came into the works. His name was Carl Muros. He postulated that there might be a link between certain population of bacteria, Clostridium difficile, Helicobacter pylori, which is this bacteria is very interesting because this is also linked with ulcers. So it's you can't really say that this is also linked to Parkinson's disease. All these are just bad bacteria that would essentially cause any other type of disease. So it's hard to just state that these bacterium are linking to PD. They're just bad bacteria that you shouldn't have in the first place. But what he was postulating is that this bacteria may cause pathology because they essentially increase the secretion of a dangerous chemical called hydrogen sulfide. And hydrogen sulfide has pathogenic mechanisms by causing the release of a certain factor from mitochondria. So it would go to the mitochondria and cause the release of a cytochrome C. Cytochrome C is essential, and when cytochrome C is present within the cytosol, and alpha-synuclein is also present, alpha-synuclein aggregation is caused by this. It causes alpha-synuclein aggregation when it's present within the cytosol. So from this sequence, Carl Morris concluded that when these bacteria are present, there's more of a chance of alpha-synuclein aggregation occurring. But the pathogenic mechanisms were still vague, and the evidence linking environmental toxins to alpha-synuclein aggregation culminating in PD pathology is starting to mount. So even though he was stating that there's a certain problem within these bacteria that are causing this hydrogen sulfide increase, they don't really necessarily they can't really be the reason why Parkinson's disease occurs. So what he's stating from here is that the pathology is most likely the pathology may be coming from something else, but in the interest of this presentation, we're going to be focused on environmental toxins, which have indeed been linked with alpha-synuclein aggregation and the pathology of Parkinson's disease. Before we get to that, we want to talk about what alpha-synuclein is, and more specifically, why is it important? Well, alpha-synuclein is an important protein. I guess it seems like it's just a bad thing to have, but it's actually a really important protein. It's mostly found within the presynaptic membrane, and is more associated with snare complex, which is a protein complex that essentially shuttles vesicles which are filled with neurotransmitter from either the extrasynaptic space into the presynaptic membrane, or vice versa. So what alpha-synuclein does is it kind of regulates this process, as well as dopamine and calcium regulation. It makes sure that the amount of neurotransmitters that are within the synapse are very little. So it does this by increasing the amount of vesicular monoamine transporters that vesicles have, so that they can kind of transport the free dopamine that may be, or free neurotransmitter that are floating within the presynaptic membrane to be shuttled within the vesicle itself, so that they can stay within the vesicle. Because when dopamine is free and just hanging around the cytosol and not within the vesicle, or not acting as a neurotransmitter on a postsynaptic membrane cell, on a postsynaptic receptor, it can cause free radical formation, because dopamine is easily oxidized when in the presence of oxygen. It can be easily oxidized, and this can rapidly lead to the accumulation of free radicals. So it's important that alpha-synuclein is present, and making sure that dopamine is not just free hanging by itself, but rather is being steadily shuttled in back to either vesicles, or vesicles that are coming from, or vesicles that are outside are being shuttled into the appropriate spaces, so that the homeostasis is maintained and just balanced properly. Another function of alpha-synuclein was that it inhibits the rate-limiting enzyme called tyrosine hydroxylase. Now, this is very interesting, actually. When you think about Parkinson's disease, we think about the accumulation of alpha-synuclein, right? Alpha-synuclein accumulating within Lewy bodies that are found in autopsies of patients who died from Parkinson's from Parkinson's disease. Tyrosine hydroxylase is an enzyme that causes the synthesis of dopamine. So the fact that this alpha-synuclein inhibits it, you can draw the conclusion that when it's increased, in the case of aggregation, when it's aggregated and there's a dense amount of alpha-synuclein within the presynaptic membrane, and then you also have dopamine, which is also free-floating. So alpha-synuclein is aggregating and not doing the normal function of increasing the amount of vesicular monoamine transporters that are there. Thus, dopamine is free-floating, causing free radical formation. And the other thing is, this aggregated alpha-synuclein is also causing an inhibition of tyrosine hydroxylase. So if dopamine is free-floating there, it's causing free radical formation. So normal dopamine is going to be synthesized because the aggregated, or the amount of alpha-synuclein that's within the presynaptic membrane is not going to cause, is going to inhibit the tyrosine hydroxylase from increasing the amount of dopamine that can be used. Thus, dopamine is just the whole dopamine homeostasis is completely disturbed because of this, as you can see here. So once PD, so this kind of just gives us a picture of what would be happening in the brain of somebody who has Parkinson's disease. So that's a problem here when it comes to alpha-synuclein aggregation. It can cause this inhibition. It normally would in normal circumstances, it will limit the amount of tyrosine hydroxylase that's present. But when it's upregulated, that limitation is also upregulated, meaning dopamine would not be synthesized at all. So another important information about alpha-synuclein is that it also exists as it exists normally as a tetramer. So that's just like four different proteins. So it has 4 subunits essentially. And another thing about it is that when it starts aggregating, this reaction is is spontaneous, in the sense that it's favorable. It's favorable such that it wouldn't want to return back to the tetramer phase that it was. So once that aggregation process starts to begin, it's very hard for it to go back to its original form. So it's something that also is detrimental to alpha-synuclein. Once that aggregation process begins, it can progress at a very, very fast rate. So the aggregation also, obviously it needs certain systems to be in place in order for certain conditions needs to be put in place for it to occur. The Lewy bodies, which are the alpha-synuclein containing protein aggregates, can be environmentally or genetically initiated, which essentially means that there could be like a toxic agent that comes in and essentially starts this cascade, or it could be genetically initiated in the sense that a certain mutation occurs in a certain gene that codes for this protein. This gene is called SNCA, and when this mutation occurs, and then this gene is transcribed, the protein product tends to come out as a misfolded state. Now, if it comes out as a misfolded state, like I stated earlier, once that sequence of aggregation occurs, and it can occur if it's not in a tetramer. So if it's not in a tetramer, then it's obviously the aggregation is going to start. So once it's transcribed, and then it's transcribed within the context of someone who has this genetic mutation, the aggregation starts to take place really, really quick, especially if it's in the context of also some form of environmental toxin that is present, thus propagating this aggregation at an earlier age and just favoring this self-assembly and maintaining this self-assembly with future transcriptions of this protein. So familial PD arises from mutations to the gene that codes for this protein. And obviously, it causes the aberrant proteins would be the misfolded proteins that are caused because of this. And these proteins are much more prone to self-assemble and much less likely to go back to the tetramer that is the original form of the alpha-synuclein. I have this slide here to kind of emphasize more of the point of more researchers more people who came to the conclusion that there is a link here between gastrointestinal disturbances and the prognosis of PD. More specifically, Brock et al. They discovered that when they autopsied the neurons or the gastrointestinal systems of those people who had Parkinson's disease, they noticed that with those who had the severe stages, they also had an increase in the amount of alpha-synuclein that was present in their gut. So this added weight to the fact that there might be a link between gut inflammation or gut disturbances linking to alpha-synuclein aggregation within the brain via the transport of the vagal afferents that are basically linked to the gut. So they essentially just put what they were stating with this specific staging was that there's a certain pathology that begins within. So the dorsal motor nucleus of the vagal nerve, this is connected to the vagus nerve, and they postulated that when the GI symptoms were increased, when the alpha-synuclein was increased within the gastrointestinal system, they would basically infiltrate the vagus nerve and then travel to the dorsal motor nucleus of the vagal nerve, and then from then on, the alpha-synuclein aggregation would just kickstart the pathogenesis that eventually becomes PD, such as But that so what they're postulating is that stage is the first stage, rather than this stage, which is what we understand now to be the beginning of substantia nigra neuronal loss. But this for them this was just a third stage. The first stage was just that those vagal afferents just essentially collecting alpha-synuclein aggregation that was occurring within the gut and just transporting them essentially to the brain, right? All right, so I've been talking about the enteric nervous system, but what is this enteric nervous system? So the enteric nervous system it's an autonomic nervous system. It's more associated with making sure that what you eat is transported appropriately from your mouth all the way to, or from your throat all the way to your, all the way to your stomach, all the way to your intestines, all the way to your anus. And the sequence of moving food within this tubular structure is called peristalsis. And just a brief overview of how the enteric nervous system would do this is, first, it would have a sensory neuron that senses the distension or the stretching of these tubular structures, and then from then on, that neuron will send impulses to interneurons that link to other neurons that would cause the relaxation and simultaneous contraction of distal and proximal ends of this tubular structure, so that this food can be passed down. So what it will do is the sensory neuron will sense that the tube is getting stretched out, and once it senses that, it will basically it would send an impulse to the end by which the food is going to basically relax, whereas the end by which the food is coming from, it will cause it to contract. So for the food to pass down, this end has to contract. So imagine this is the food. This end will have to contract, right? So it can force the food to go down. But the food is not going to go down if this end is also contracting. So this is going to contract, and this part is going to relax so the food can come down. And then the nerve that is linked to that area would sense that, and the nerve that food essentially moves onto would sense that distension, and then it would propagate the same kind of impulse. It would cause the simultaneous contraction and relaxation of distal and proximal areas, so that the food can be passed down appropriately. But more specifically, I don't want you to care. You don't need to focus on this specific everything on this image. I just want you to focus on my enteric plexus, which is more important to what I'm going to be talking about next. And what the enteric plexus is, it's a nerve bundle, nerve ganglia essentially, of the enteric nervous system that that helps with this peristalsis movement. So it's more associated with moving the food bolus within the small intestines and the large intestines, by contracting the muscles that are associated with where that food is kind of moving. So it's responsible for peristalsis movement within the gut. Although it can work independently, these fibers also receive innervation from dorsal motor nucleus of the vagus. So that's how this connection occurs. So it's basically its own brain, right? Because it's just a system of neuronal ganglia that are just contracting muscle fibers. But it can receive some form of innervation from the dorsal motor nucleus of the vagus nerve, such as like this innervation can come in the sense of like when you have when you're nervous, for example, you kind of feel like butterflies in your stomach, right? So that the anxiety is occurring within your brain, but when you have those butterflies, when you have that butterflies feeling, that synapse, it's sent in by the vagus nerve. That's the vagus nerve just kind of like communicating with the vagal with the vagal efferents that connects to the enteric plexus, and just kind of cause some small contraction of muscle that kind of convey that feeling of like butterflies in your stomach type thing. So that's just the enteric nervous system at work, basically, and that's how it's basically connected to the brain in its own way. That's basically the only way that the brain would innervate when talking to the enteric nervous system. But moving on to what I was talking about earlier, and when it comes to like environmental toxins and the development of Parkinson's disease of unknown origin, or idiopathic PD. Rotenone is an insecticide that has been shown to have an abnormal release of alpha-synuclein. So rotenone was administered to some mouse models to cause Parkinson's disease, and what they wanted to do was they wanted to basically see whether they can attenuate this. So what they did is they had some mouse models. They kind of some of them got rotenone, and then some of them didn't for control purposes. And within the subsets that received rotenone, some of them had their parasympathetic and sympathetic nerves that are there. These are just like basically they vagotomized, or they cut the nerves that link the enteric nervous system to the vagus nerve. They cut those nerves, and then they wanted to see whether they could stop the pathogenesis of Parkinson's disease. What did they find? They found that those who had the cut nerves, even though they had an abnormal secretion of alpha-synuclein or an abnormal accumulation of alpha-synuclein within their gut, they did not have PD. But those who didn't have their nerve skirt, they eventually developed Parkinson's disease, which led them to conclude that there is a link between some environmental toxins and the pathogenesis of Parkinson's disease. This is just one of those papers that was stating that, and I just wanted to put that out there. But yeah, so finally, let's get to the paper that is of interest here. So this paper, what is it saying? Ingestion of subthreshold doses of environmental toxins induces ascending Parkinsonism in rat. So this means that when they some small doses of some type of environmental toxins may cause Parkinson's disease. Their hypothesis was that when they introduced subthreshold doses of a certain toxin called Paraquad and lectin, these lectins are plant toxins, they provoked an ascending pattern, or rather an increasing accumulation of alpha-synuclein accumulation within the vagus nerve and the dorsal motor nucleus of the vagus nerve, or the vagal nerve. And with also they noticed there was consequent gastric motility. This means that there was a disruption with there's a disruption within gastric. So there was a disruption between how the enteric nervous system was able to communicate with the smooth muscles of the gastric system, and also subsequent degeneration of substantia nigra, pars compacta neurons, and eventually causing idiopathic PD. So these toxins. I'm just going to talk about them here briefly. Paraquad is a very common herbicide. It's actually still used in the United States. It's used in farming usually. It's an acute exposure to this toxin can eventually lead to death. But most commonly, people who kind of live in this kind of like farming communities are more at risk of developing diseases related to Parkinson's disease when they have frequent low dose exposures to this toxin. Lectins are carbohydrate binding plant toxins that are usually found in raw uncooked foods such as legumes, eggs, and grains. The reason why they use lectins here is because they think they use lectin as a driver to essentially kind of bring the paraquat to the brain, because it has the capacity to bind to carbohydrates, and carbohydrates are just carbohydrates are they're moieties that are kind of sticking out of moieties that stick out in certain cells, such as the intestinal cells that line the intestinal tract. They have moieties of carbohydrate moieties that are just sticking out, and lectin has the capacity to attach to those moieties, and then essentially just kind of open a gap and then enter into the circulation, causing destruction in that way, because it's a plant toxin. So if what they think is that if they introduce paraquat and lectin, this is the sequence by which Parkinson's disease may occur, or rather there's a possibility that this can lead to Parkinson's disease. So what did they do? They created a solution of lectin and paraquat, and then they fed it to Sprague dolly rats for about 7 days. And then they administered a type of motor test called whisker-evoked forelimb test and stepping test to check whether they had Parkinsonian type symptoms. So these tests just kind of involved restraining the rat, the forelimbs, the hind limbs of the rat, and trying to see whether it was able to move the forelimbs by itself. Rather, another group of rats received a vagotomy. What this means is they received a resection, or they cut the vagus nerve, preventing paraquat from preventing alpha-synuclein from aggregating, or from not aggregating, but preventing alpha-synuclein from transversing into the vagus nerve. And another group as well received some levodopa treatments to see whether they could attenuate or stop the degeneration that the alpha-synuclein was causing. So they wanted to see whether they could kind of model the same kind of treatment mechanism that humans usually receive. So whether this was the same kind of Parkinson's disease that is related to humans who receive levodopa treatments. The gastric motility. The reason why they did this is they wanted to see whether gastric motility is the degree by which the enteric nervous system is able to contract the stomach, essentially. So they tested. They put some string gauges, and then they sutured them onto the stomach, the upper surface of the stomach, and they basically took recordings of it. They administered a muscarinic agonist to make sure that there was no impairments and no confounds that may have led to impairments of the smooth muscles. They wanted to make sure that the smooth muscles worked appropriately when they tested it by administering certain agents within the brain to promote this gastric motility. So they wanted to see whether once they administered the toxin, once they administered the toxin, the gastric or the movement of the stomach lining was appropriate, because it was affected because of the treatment and not because of some other reason that the smooth muscles were just messed up because they were for some reasons unrelated to the experiment. Essentially, that's what they kind of administered that muscarinic agonist to just see if the smooth muscles were working properly. Alright, so what are the results? So at first they wanted to see if alpha-synuclein does aggregate in vitro, and they saw that it does actually do that once they introduce appropriate mechanisms. And as you can see here, the aggregation was steadily increased. But as compared to paraquat and lectin by itself, once they introduced Paraquad, the aggregation wasn't as it was much more increased actually. So with Paraquad, the aggregation was much more increased. There was more aggregation, just pointing to the fact that if you introduce Paraquad, it's going to cause aggregation. But when they introduced Paraquad and lectin, as you can see here, the time it took for the aggregation to occur was much more less. So the half time was significantly decreased. So here, Alpha-Synuclein aggregation, it took about 27 hours for the aggregation to occur. But when Paraquad and lectin were added, the aggregation took about, I would say, about 17 hours. So much more less time it took for that aggregation to occur. And here for the motor test, when they administered, so here we have the first test. This is the whisker test. Excuse me. The whisker test here, they noticed that at baseline, the baseline they almost performed perfectly, but at 2 weeks after the treatment of Paraquad and lectin, the performance was severely decreased, and as you can see here, it was even furthermore decreased at 4 weeks. And once they administered L-Dopa, there was a slight increase in activity in gain of function again, which just indicating that this was the same type of sequence that is of Parkinson's disease that we see typically. Here we can see that when they administer the treatment and they cut the vagotomy, as they performed the vagotomy, so they cut the vagus nerve, they noticed that there was also a increase in there was no change in how they were able to perform motor even after introducing after introducing these toxins. The same for the steps here. As you can see, the baseline was great. After 2 weeks and after 4 weeks, it was much more increased. And with L-Dopa, it seems that L-Dopa just stopped that decrease. And when they cut the vagotome, or when they performed the vagotomy, it was also saved as well. And in this one, in this column here, this is the stomach lining, and this is the small intestine, and this is the large intestine of the rats. This is the control, and this is 2 weeks, 4 weeks, and then this is the rats that received the resection or the vagotomy. So in these ones, you can see the green indicates, it shows the amount of the amount of alpha-synuclein that was present within these cells. As you can see, it was much more increased in this 2 weeks, and compared to the 4 weeks here in the large intestine. Look how green it is in the large intestine. You can see those significant aggregation after 2 and 4 weeks of toxin treatment. Same goes for this one. I mean, there's barely any here because this is the stomach, right? So the stomach, small intestine, large intestine. Once the vagotomy was produced, even though there was indeed some fluorescent, there wasn't really. I mean, there was alpha-synuclein aggregation because of the treatment. This is basically what that is stating. So moving on to this one, actually, this is the brain area of the brain areas that were of interest. When it came to the alpha-synuclein presence of alpha-synuclein presence within the brain areas. So the brown is basically tyrosine hydroxylase positive neurons. So these are the neurons that basically produce dopamine. And this upper row here is the brown. The upper row represent the control animals. You can see here the brown is very prevalent, especially in the substantia nigra pars compacta, very, very prevalent. And this is 2 weeks after, 2 weeks after the introduction of the treatment. As you can see, it was much more decreased here. The brown was very, very much more decreased, and even much more so in the 4 weeks following that. Same as you can see here too. The decrease was much more prevalent within the 4 weeks after the treatment mechanism was given. This is kind of stating the same thing here. This is a control animal, and this is the animal that received the treatment in the substantia nigra pars compacta. As you can see here, the stain is much more rich, but here it's very, very stark. It's very much more less decreased, owing to the alpha-synuclein aggregation that occurred. And essentially this one, it's looking at the treatment 4 weeks after. Within the left substantia nigra pars compacta, and the right substantia nigra pars compacta was there's more degeneration here in the right, as you can see. And in this one was testing for the amount. This is just kind of like a test of whether the tyrosine hydroxylase was lost because of neurons. CV was testing the amount of neurons that were lost, and this was just looking at the amount of tyrosine hydroxylase. So they used this test to make sure that the loss of tyrosine hydroxylase was not due to just some environmental or subjective loss of downregulation of tyrosine hydroxylase, but rather it was a loss of the neurons, and they used this as a gauge to see that the loss of neurons was still very, very prevalent. And in this test, they tested the gastric motility. As you can see here, this is the control animal. So when they administered the NMDA within the substantia nigra pars compacta, what you would expect is the recordings that were done by the strain gauges were they were producing some very beautiful peaks here, as you can see. So this is how it should perform normally. But with the treatments, 2 weeks and 4 weeks, you can see there was a significant diminishing of those peaks, especially after 4 weeks. In this one, it's kind of saying the same thing here. It's testing the tone, whereas this one's testing the motility. As you can see here, at 2 weeks and at 4 weeks, there was a significant reduction of function here. This one is kind of saying the same thing here. So they introduced tyrosine hydroxylase or thyrotropin releasing hormone at control. As you can see here, it was beautiful. It was showing some beautiful peaks, and the peaks just steadily were decreasing at 2 weeks and 4 weeks. And this is when they introduced different concentration of thyrotropin releasing hormone in 0.1, 1, and 3. This is from the control animals. As you can see, the motility was increased as the dose of the thyrotropin releasing hormone were increased. And at 2 weeks, you can see there was still a significant decrease, and even more so at 4 weeks. And the same goes here for the gastric tone as well. Reaching on to the toxin treatment, or reaching on to the discussion, it's possible that the toxin treatment did produce Parkinsonian-like pathology that basically began in the gut, and this progression was caused by the aggregation of alpha-synuclein within the ENS prior to entering the dorsal motor nucleus of the vagus nerve, and then progressing to the substantia nigra, which basically led to what they were expecting, which is that there was significant reduction in alpha-synuclein progression following subdiaphragmatic vagotomy. So when they did the vagotomy, they noticed that the amount of vagal, once they cut the vagus nerve, they basically saved a lot of the neurons, right, of the substantia nigra. So to conclude, the experiment justified that the authors were right, that the Paraquad and Lectin treatment that was given to the rats led to the PD pathology that we saw, and was saved by administration of L-DOPA therapy, as we saw early in the results. And they also noticed that motor symptoms were ameliorated once the vagotomy was performed, even if the Paraquad and Lectin treatment was performed. So that leads to the end of my presentation. Thank you very much for watching, and I hope